Hello everyone, I am Harun. In this video we are going to discuss about the identification and cultivation of algae. First let's look at algae. It is protist and plant-like characteristic. It is found in aquatic environment. It is eukaryotic organism. It contains chloroplast. Chloroplasts are plant cell organelles that convert light energy into relatively stable in chemical energy. It capable of photosynthesis. It produces own foods. It have no roots, stems or leaves. It can be seen both microscopic and macroscopic method. The algae reproduce by two different methods, namely sexual and asexual method. Next, identification of algae. Collection method. Let's see how to collect algae. Micro and macro algae are collected using knife or by hand. To these different reproductive cells, often a pocket microscope is used. This is important for some genera. Microalgae are organisms that can grow in aquatic environment and use light and carbon dioxide to create biomass, and macroalgae, commonly called seaweeds and marine plant that photosynthesis but reproduce without flowers. Macroalgae are multicellular large-sized algae visible with the naked eye, while microalgae are microscopic single cells and may be prokaryotic similar to cyanobacteria. For example, phytoplankton is collected using mesh net. Phytoplankton is a marine algae. It is also known as microalgae. Then water samples are kept overnight to settle or concentrate the algae towards the bottom. In some cases, algae samples are sequenced to get more biomass, like in the case of spangium. Samples that are present in soil are difficult to collect, therefore, the culturing before the microscopic examination is done to get sufficient biomass. Some data should be recorded before the next step of identification. First, first one is name of collector, second one is data of collection, third one is locality of algae, fourth one is the texture of water from which algae is collected, fifth one is algae either free floating or attached. There are three types of macroalgae present in the world: red algae, brown algae, green algae. Red algae scientific name is Rhodophyta. Brown algae scientific name is Pyophyta. Green algae scientific name is Chlorophyta. Next, microscopic examination of algae. First, a clean glass slide was taken. Place a drop of water or specimen on a microscopic slide containing algae. Gently cover the slide with a cover slip and view under a microscope. Be sure to use a specimen of very small quantity to avoid clumping. Observe slide in the microscopic range of about 40x or 100x magnification. The hanging drop method can also be used for microscopic examination, in which the sample is taken on a cover slip and paraffin wax or liquid paraffin is used to examine. The hanging drop technique is a well established method for examining living unstained very small organisms, and using a compound microscope, a higher power greater than 100x would be necessary to be properly observe specifics. Drawing or photographs are essential for algae to be properly identified. For this, a camera attachment can be used. Scanning and transmission electron microscopes are rarely used for this purpose. They are only used for a few very small algae. And then cultivation of algae. Microalgal culture were achieved by Bajornek in 1890 with Chlorella vulgaris. Chlorella vulgaris are green single-celled microalgae. Cultivation of algae, also known as algal farming or algae cultivation, is the process of growing and harvesting algae for various purposes including food production, biofuel generation, wastewater treatment, and environmental susceptibility. Algae are simple photosynthetic organisms that can thrive in a wide range of aquatic environments from freshwater to saltwater. Adds an overview of cultivation of algae. First one is select the algal species. Choose the specific species for algae you want to cultivate. The choice of species depends on your goals, whether it is for research, biofuel production, food production, cosmetics, or any other application. Second one is cultivation system. The two most common methods of microalgae cultivation are open cultivation system such as open bonds, tanks, and controlled closed cultivation system. An open cultivation system. Algae can be grown in open bonds, typically shallow and large, where they receive sunlight directly. These systems are relatively low cost but are susceptible to contamination and require a lot of land. Closed photobioreactors. These are enclosed systems that offer more control over environmental conditions such as temperature, light, and nutrient levels. They are more expensive but provide a higher degree of productivity and reduce contamination risk. These are two types of cultivation system. First one is open cultivation system. Second one is closed cultivation systems. Third one is nutrient supply. Algae need nutrients including nitrogen, phosphorus, and trace elements for growth. These nutrients are typically supplied in the form of fertilizer or through wastewater treatment processes. Light sources. Algae require light for photosynthesis. In open bond system, they rely on natural sunlight, while enclosed systems use artificial lighting to control and optimize the light exposure. Microalgae require light to produce ATP and NADPH and synthesize essential molecules for growth. Fifth one is temperature control. Maintaining the right temperature is essential. Algae have specific temperature requirements depending on the species. In closed system, temperature control is more manageable. When the temperature is below 15 degrees Celsius, microalgae growth will slow down, but when the temperature is higher than 30 degrees Celsius, microalgae growth will stop. Sixth one is harvesting. When the algae reach their desired biomass, they need to be harvested. Various methods are used, including mechanical filtration, sedimentation, and centrifugation, depending on the cultivation system and application. Algae harvesting is the process of cultivating algae for use as biofuel. The commonly used harvest method is through gravity settlement or centrifuge. The oil from the biomass will be removed through solvent extraction and further processed into biodiesel. Seventh one is processing. Once harvested, the algae can be processed for their intended use. This could be involve drying, cell disruption, extraction, and purification. Thank you.